Hello, this is DNA Replication by Austin Tipper. Here we have a parent DNA strand. Now, what happens is this parent DNA strand will divide into two daughter DNA strands. Now, this is what a DNA strand looks like unraveled. You have a 5' and a 3' end. And what happens is you get these ACTGs. And what happens is, on the other side, you're going to get TCGA, which would make the bottom go 3, 5. And this is what we call an anti-parallel structure. Now, these A, G, C, and T's are the nitrogen bases, which code for the building of proteins. So the order they come in is very important. Now, once the strand's built, this primer have to be removed, and then the nucleotides are filled in. And what has to happen is a DNA ligase has to come in and join these two bits together because it's not connected. Now what happens is at the end of the DNA strand, you end up getting this scenario, where you have this area that can no longer be replicated because there is no longer room because it forks together. So no more DNA must come in. So what happens is it ends up getting cut off, losing nucleotides and potentially losing valuable DNA information. What you'll see is this thing called telomerase will actually come in and attach telomeres, which is this area that is okay to be cut off at the end of DNA replication because no important genes are there. What this allows is DNA, every time it's replicated, to be shortened just a little bit without actually losing any genetic information. And so once that's done, you have two new DNA strands that, if the telomerase did its job right, have all the DNA necessary for life to live on and the DNA to replicate again. Thank you. This was DNA Replication with Austin Tipper. Goodbye. So this anti-parallel structure is very important that it doesn't end up getting messed up while it's being replicated. Now what's going to happen is we're going to have this thing called a helicase come along, and what its job is to cut the DNA in half down the middle so we can have these two separate strands. What you'll also see is a single strand binding protein, or SSBP, and what its job is to hold this gap apart before DNA replication can happen for this strand. Now what we end up getting is this thing called a DNA replication bubble, and that's this big thing in the middle. And on either end you have a DNA replication fork, which is basically where a Y is created, where the two strands are cut apart. What you have in the middle of all of this is the origin point. And what this is this is the original spot where the two helicases came together and started unwinding the DNA strand. Now what will happen is we'll get a leading strand and a lagging strand. And we'll explain what those do later. What we'll see is this primase will attach a primer to the DNA strand. And what's important about this is it creates another 3 and 5 end, which is important because DNA replication can only go in one direction. And what this prime area is made out of is RNA. The important thing is that DNA polymerase can only add to the 3 side of the nucleotide. So it can only add new nucleotides on this side, here, here, and here, or here, here, and here. And what this does is it will create the leading strand and the lagging strand. Because this leading strand is going to be able to continuously create more DNA without having to slow down. But this lagging strand, on this lagging strand, what you're going to see is they're going to be built on this side of the primer, creating an Okinawa, sorry if I butchered that name, strand, which is the segment that's being built. And what happens is the primase, DNA polymerase, will start at one spot, build here, move this direction, and once it hits this other RNA strand, it has to jump all the way back and start building here again.